Observe the rapid expansion of these floral structures. Within seconds, every component moves into a specific, predetermined alignment. These seed-bearing plants, categorized as phanerogams, rely on the flower as their primary mechanism for sexual reproduction. Botanically, a flower is classified as a modified shoot, specialized for the complex tasks required to produce the next generation. The biological objective is the successful achievement of fertilization, leading to the development of viable fruits and seeds. To understand this process, we must deconstruct the distinct anatomical architecture of what is known as a complete flower. This structure functions as a multi-stage factory designed to protect fragile genetic material while ensuring its successful transfer. The morphology of the flower is organized into 4 primary concentric whorls. Species like the hibiscus, which possess all 4 layers, are complete flowers. In contrast, plants like the pumpkin produce incomplete flowers, lacking 1 or more of these whorls. We model this system by building it from the base upward, beginning with the physical anchor. The primary stalk, or pedicel, connects the floral apparatus to the main stem. At the apex of the pedicel, the structure widens into a swollen platform called the thalamus. The thalamus serves as the central receptacle where every successive whorl is securely mounted. This platform provides the necessary structural integrity for the delicate reproductive systems that sit above it. The first layers are the accessory whorls. These do not produce gametes, but instead manage defense and the logistics of pollination. The outermost envelope is the calyx. Composed of individual units called sepals, the calyx acts as a physical shield, protecting the flower while it is still in the bud stage. Because sepals contain chlorophyll, they also contribute to the plant's energy production via photosynthesis. Internal to the calyx is the second whorl, the corolla. The corolla consists of petals, which utilize specific pigments and scents to attract pollinating vectors like insects. These petals also provide a secondary layer of protection for the reproductive organs housed at the center. The accessory whorls function as a perimeter defense, neutralizing external threats so the inner core can focus on the production of reproductive cells. Moving further inward, we reach the first of the 2 strictly reproductive layers. This third whorl is the androecium, the male reproductive system of the flower. The androecium is made up of individual units known as stamens. A single stamen consists of a slender stalk called the filament. The filament dictates the spatial positioning of the reproductive head, ensuring it effectively contacts visiting pollinators. At the top of the filament is the anther. The anther is the manufacturing center where microspores, commonly known as pollen grains, are synthesized. The androecium is essentially an elevated factory designed to export male genetic material with maximum efficiency. At the absolute center of the flower sits the final structural component. This innermost core is the gynecium, which is built from foundational units called carpels. The gynecium is divided into 3 zones, starting at the top with the stigma. The stigma is the receptor pad, specifically designed to capture incoming pollen grains. Below the stigma is the style. This tube acts as a secure transit corridor, leading from the point of capture down to the base. The third zone is the ovary, the swollen chamber at the base of the carpel. The ovary provides a shielded environment for the ovule housed within it. Following fertilization, the ovary matures into the fruit, while the internal ovule develops into the seed. The gynecium is the system's final destination, an armored incubator positioned at the heart of the flower to ensure the survival of the next generation. Viewing the reassembled model, we see how these layers function as a single integrated system. The flower's success depends on this precise arrangement, balancing the physical protection of internal organs with the logistical requirements of pollen transfer. This concludes our analysis of the macro-level morphology of the angiosperm. While this mapping explains the macro architecture, the actual manufacturing of genetic material happens at a much smaller cellular scale. In our next session, we will look inside the anther to observe the mechanics of microsporogenesis. Understanding the structure of the flower is the prerequisite for unraveling the microscopic engineering occurring deep within its internal factories.